You know, we have this idea that to build something strong, you have to be careful, right? Gentle, precise. But what if nature's most incredible designs uses strategy that's, well, the complete opposite? What if the real secret to creating something powerful is to first break it? I know, it sounds totally backward. From the time we're kids, we're taught breaking things is bad. It's the end of the road. But in this explainer, we're going to see how biology completely flips that idea on its head. And look, that makes perfect sense in our everyday world. Think about a crack spreading across your phone screen, or a fault line in the earth. It's a sign of weakness, a structure that's giving up under stress. It feels final, doesn't it? Catastrophic, even. But here's the twist. In living things, a fracture isn't always some random accident. Scientists are now discovering it can be a super-controlled, precision-engineered event. It's a fundamental tool that evolution uses to sculpt and build some of the most amazing structures imaginable. So where better to start this story than at the very, very beginning of life? We're going to look at a mouse embryo just a few days after conception in what is one of the most delicate and crucial moments of its entire existence. Just look at this. It's incredible. We're seeing a tiny ball of cells that's on its way to becoming a mouse. And to do that, its first big job is to hollow itself out. It literally pries its own cells apart with fluid to create this cavity, the very space where the fetus will eventually grow. It's like a controlled demolition to make room for new life. So how does this microscopic construction project actually work? Well, it's a 5-step dance. First, the cells start to pump fluid into the teeny tiny spaces between them. Second, this creates hundreds of little bubbles that start to expand, pushing on everything around them. Third, that pressure causes a fracture, literally prying the cell membranes apart. Fourth, all those smaller bubbles start to merge into bigger ones. And finally, boom, they create one single hollow cavity. The whole thing has just transformed itself. And this merging of bubbles, it's not some biological magic. It's pure physics. It's a thing called Ostwald ripening. And you've seen this happen before. Think about a bubble bath. After a few minutes, what happens? All that fine, dense foam disappears, right? The little bubbles have emptied their air into fewer, bigger ones. That's it. It's just pressure differences. And that exact same physical law is running the show inside this tiny embryo. Amazing. And here's where it gets really clever. This whole process isn't just guided by genes, it's dictated by physics. See, not all the cells in that little ball are identical. Some are kind of weaker, they deform more easily. Others are tenser, with more robust internal scaffolding. So what does the fluid do? It takes the path of least resistance, pushing apart the weaker cells to form that cavity, while the tenser cells hold the whole structure together. It's so effective that, as researcher Harvey Turlier puts it, the physics of the situation is so dominant that the embryo has no other choice. And this is what makes the process so incredibly reliable. By just letting simple mechanics do the heavy lifting, nature makes sure this absolutely critical step in development happens perfectly every single time. Okay, so a controlled fracture works in a delicate, tiny embryo, fine. But what about in an organ that's built to withstand immense, constant force for an entire lifetime? Let's take a look at an even crazier example, the developing heart. I mean, the contrast here is just wild. We're going from this gentle hollowing out process to building the most powerful muscle in the body. How could a process that literally involves breaking things possibly work in a place where strength and durability are the entire point? To get a sense of the forces we're talking about, check this out. A developing zebrafish heart, which is a great model for our own, beats 150 times a minute. And that's not after it's built. That's while it is still being built. Can you imagine trying to construct a skyscraper during a constant, powerful earthquake? That's the level of mechanical stress we are dealing with. And here is the beautiful paradox. The very force that you'd think would tear this tiny heart apart, its own relentless beating, is actually what forges its intricate internal structure. The constant strain isn't a problem to be solved, it's a key part of the construction process. And what is it building? These things called trabeculae. You can see them here, this complex, web-like mesh of muscle inside the heart. Now, these aren't just for decoration. They are absolutely critical for a powerful, efficient heartbeat. Without them, the heart just can't pump blood the way it needs to. So how does the heart use its own force to build itself? It's incredible. First, all that beating concentrates strain on the outer wall. This causes the internal scaffolding, this stuff called cardiac jelly, to weaken and literally break. This creates these big gaps, actual fractures in the jelly. Then, heart muscle cells fall into those cracks and seed themselves there. And finally, those cells link up and build the trabeculae. The heart literally breaks itself to become stronger. So we've seen a delicate embryo and a powerful heart, both using fractures to build themselves. But these aren't just a couple of weird, isolated quirks of nature. Nope. They are star examples of a much bigger idea, a field of science that is basically rewriting our understanding of how life gets built. And that field is called mechanobiology. It's the study of how physical forces, pushing, pulling, straining, and even breaking, aren't just side effects of biology, they are core instructions in the blueprint of life. Understanding these forces is huge because when they go wrong, they can lead to disease. And if we can master them, it could totally change how we engineer and repair our own bodies. This really is a massive shift in thinking. The old view was that our genes were the complete blueprint, a set of instructions that dictated every single step. But the new discovery is that physical forces are a co-architect. In fact, some researchers have found that these mechanical processes can happen, quote, too fast for the genome to play a role. Basically, physics takes the wheel when the genetic code leaves off. So what does all this mean for the big picture? It means that this whole break it to make it strategy, it's not a fluke. It's a powerful, super efficient, and surprisingly common strategy that nature has been hiding in plain sight this whole time. And once you start looking for it, you see it popping up everywhere. This isn't just for embryos and hearts. Scientists are now finding that constructive fracturing helps shape the nostrils of zebrafish, the mouths of hydra, the actual joints in the legs of fruit flies, and it even helps an entire flatworm regenerate from just a tiny piece. It's a universal tool in nature's toolbox. Rashmi Priya, one of the scientists who studied the zebrafish heart, just sums this whole thing up perfectly. She says, in biology, breaking isn't always a failure. It's often a necessary step in building something new. That one sentence completely reframes how we should think about how life is built. And that kind of leaves us with one last big question to chew on. This whole discovery shows us that one of our most basic assumptions about creation and destruction was totally backward. So if nature's own blueprint for building strength actually starts with a crack, what other fundamental ideas about how the world works might we have completely wrong?